Antibody classes. IgG subclasses include IgG1, IgG2, IgG3, and IgG4. It's the only immunoglobulin that crosses the placenta. IgM, produced in secondary or anamnestic antibody response. IgG1, IgG2, and IgG3 activate the classical complement pathway. It is the predominant serum antibody, making up approximately 75% of immunoglobulins in the blood. 5 monomers are linked together by a J chain and interchain disulfide bonds. It constitutes about 10% of total serum immunoglobulins. IgA. It is the first antibody produced against an immunogen, produced in both primary and secondary immune responses. IgM is the best activator of the classical pathway of complement, with only one molecule required. Serum and secretory forms. Serum IgA is a single immunoglobulin molecule, whereas secretory IgA is a dimer held together by a J chain. It accounts for 15 to 20% of total serum antibody. IgD, primarily a cell membrane surface component of B lymphocytes. It has a short half-life of about 2 to 3 days. The functions of serum IgA are antigen clearance and immune regulation. The function of IgA in mucous membranes is to block the attachment of viruses, bacteria, and toxins to host cells. It is responsible for allergic or type 1 hypersensitivity reactions. The Fc portion binds to receptors on mast cells and basophils. Once attached to mast cells, IgE binding an allergen triggers degranulation of the cell and the release of allergic mediators such as histamine and leukotrienes. Elevated IgE concentrations are often found during parasitic infections. Monoclonal antibodies. They are identical antibodies that are produced from a single clone of plasma cells. These antibodies are found in individuals with multiple myeloma. Monoclonal antibodies are also produced in industry by fusing an antigen-sensitized splenic B lymphocyte with a non-secreting myeloma cell, thus creating an immortal cell line that secretes an antibody of a single idiotype. Quantification of antibodies. The purpose is to provide information about the functional immune status of an individual. IgG, IgM, and IgA are quantified using radial immunodiffusion, nephelometry, or turbidimetry. Myeloid cells. They are responsible for the nonspecific response. This includes monocytes and macrophages. In the peripheral blood, this cell is a monocyte. In the tissue, it is a macrophage. Tissue macrophages include alveolar macrophages, Kupffer cells in the liver, and astrocytes and microglia cells in the nervous system. Their functions are quite vital. They perform phagocytosis of invaders, present immunogens to T helper cells, which is the first step in an immune response, and release cytokines, also known as monokines, that affect other cells' activities. Macrophages have major histocompatibility complex class I, complement, and antibody Fc receptors on their surface. Granulocytes are a type of white blood cell that play a crucial role in our immune system. Let's dive into the different types of granulocytes. First, we have neutrophils, also known as polymorphonuclear cells or PMNs. They make up 60 to 70% of white blood cells in circulation. Their primary function is phagocytosis, and they also contribute to the inflammatory response. Next are eosinophils, which account for about 1 to 3% of circulating white blood cells. They are key players in mediating the IgE allergic response. Lastly, we have basophils. They comprise 0 to 1.09% of circulating white blood cells. Basophils have receptors for IgE and contain granules that are responsible for allergic reactions. Lymphocytes, which make up 20 to 40% of circulating white blood cells, play a crucial role in our immune system. B lymphocytes, or B cells, constitute about 20% of circulating lymphocytes. They express surface molecules such as CD19 and CD20. After birth, B cells mature in the bone marrow. These cells then differentiate into either a plasma cell, which produces antibodies, or a memory B cell. T lymphocytes, or T cells, make up 80% of circulating lymphocytes. They express surface molecules such as CD2 and CD3. Their functions are multifaceted. Cytotoxic T lymphocytes, or CTLs, lyse host cells infected with viruses and tumor cells and also produce lymphokines. T cells can stimulate, through T helper cells, or suppress, through T suppressor cells, the activity of other cells. Pre-T cells originate in the bone marrow and fetal liver, and then they migrate to the thymus to mature. Additionally, natural killer cells, or NK cells, are slightly larger than T or B cells and contain cytoplasmic granules. Other cells that assist in the immune response include dendritic cells, which present antigens to T cells. Langerhans cells, a type of dendritic cell found in the dermis and squamous epithelia, also play a role. Lastly, mast cells, which are granulocytes resembling basophiles, contain many chemicals that affect the immune response.